All right, our last speaker of this session is Anurag Agrawal. Anurag obtained his bachelor's and master's degrees from the University of Pennsylvania and his PhD from the University of California at Davis. After a postdoctoral fellowship at the University of Amsterdam, he went on to a faculty position at the University of Toronto and then was recruited to join the faculty at Cornell University, where he is now an associate professor. His research interests are broad and mainly focus on the ecology and evolutionary biology of herbivory and plant defenses.
Thanks a lot, Jessica, and thank you all for hearing me talk, and it's a pleasure to be here. It's exciting. It's already been really exciting. I guess I sort of view my role here today in a couple of different ways, and one of the things I want to do is infuse a little bit of natural history into our very abstract discussions, or at least my perception of very abstract discussions that we've been having. And as a plant evolutionary ecologist and worm-counting biologist, extend some of the discussions we started this morning about genetic correlations and trade-offs to really thinking about the specific traits and the ecological context in which we might expect different strategies to evolve or particular traits to trade off. My co-authors on this talk are Jeff Connor from the Kellogg Biological Station and Sergio Rasman, a postdoc in my lab. And before I sort of get all into it, I wanted to relay a story. As a graduate student at UC Davis, I lived around the corner from Ledyard Stebbins, and I visited him once, and he really, both his writings and that one conversation I had with him, really influenced my own thinking about the organismal strategies that have evolved and why particular organisms might show alternative strategies. And the other guy, and this is just kind of amazing, he would bicycle by my house every day, was the great tomato breeder Charles Rick. And I once listened to a seminar of his after he was talking about the breeding of frost tolerance. And at the very end, he was asked why we eat flavorless tomatoes. And after mumbling something about trade-offs for a moment, he relayed the story of when he was asked to breed tomatoes that would withstand transportation across the country, he stood on one side of a street that I knew very well with a pile of tomatoes, and he threw them to the other side. And after a couple of years of choosing the tomatoes that didn't burst open when they when they landed on the other side, he arrived at the answer of why we eat flavorless tomatoes. And so, but this talk is really about trade-offs and thinking about the specific traits that might evolve in plant-animal interactions in particular, and why alternative strategies might evolve. The concept of trade-offs to me is at least deceptively very simple. As Doug defined it, I believe, here in 2001, the advantage of a genotype in one context is accompanied by a disadvantage in another context. And I certainly can't find anything wrong with this definition, although I will say that it is still a very large problem that is not easy to define. And the way my own thinking about trade-offs has led me to think about a kind of one-trait trade-offs versus two-trait trade-offs. A one-trait trade-off involves a single trait where there's opposing or stabilizing selection on that trait. You might think the classic example is gall size on goldenrods. The fly makes a gall, and the birds prefer to eat the big ones, and the parasitoids, which are limited by their ovipositor length, prefer to parasitize the smaller ones. And so there's a trade-off there implied because the fitness in different environments, in this case, of galls are opposing. There's then the two-trait trade-off, which we might imagine as two traits being favored, but that share some limiting resource. And if there's truly a limiting resource that can only be allocated to one side or another, then indeed both traits will not necessarily be expressed. And I think one thing I'd like to come to at the very end of the talk is whether we really believe in constraints in the sense that can limiting resource. Of course, in the trivial sense, it has to be the case. But for two particular traits, will it be the case that there are inevitable trade-offs, or can selection always, if it's imposed strongly enough, get around those two? The last bit here that I want to just definitionally sort of go over is thinking about trade-offs in terms of the definitions above and distinguishing them from what we might call adaptive negative correlations. There are many negative genetic correlations, and this certainly pertains to the to the two talks we heard earlier in the morning, which may be negative, but may not necessarily represent a limiting resource or opposing natural selection. In fact, there may be negative genetic correlations that are in fact shaped by natural selection. And totally phenomenal quote from Darwin from The Origin of Species that I want to read you in terms of his understanding of trade-offs and also of this distinction between a trade-off driven by resource limitation versus by adaptation. So as Goethe expressed it, in order to spend on one side on one side, nature is forced to economize on the other side. I think this holds true to a certain extent with our domestic productions. If nourishment flows to one part or organ in excess, it rarely flows, at least in excess, to another part. Thus, it is difficult to get a cow to give much milk and to fatten readily. He also noted, I took out a bunch of the examples, the same varieties of cabbage do not yield abundant and nutritious foliage and copious supply of oil-bearing seeds. That's certainly what would be indicative of a trade-off, presumably driven by resource limitation. But what he goes on to say is the remarkable bit, which to me really blew my mind. But I see hardly any way of distinguishing between the effects on the one hand of a part being largely developed through natural selection and another adjoining part being reduced by the same process or by disuse. That's the adaptive negative correlation. And on the other hand, the actual withdrawal of nutriment from one part owing to the excess of growth in another and adjoining part. And that's what we think of as the classic resource-limited trade-off. And I'll sort of give it away now. I think that we're still a pretty far ways away from being able to distinguish the adaptive negative correlation from the trade-off. And perhaps it's not important, and that's one of the things that I think perhaps we could pick up as a thread at the very end. Maybe that distinction is not really important, but my sense is that it's still a really difficult thing to distinguish. So my worldview about trade-offs sort of looks something like this, that there are, of course, limiting resources that must be allocated to many traits. I think one of the reasons life history theory has been so successful at demonstrating trade-offs is that the traits we're thinking about encompass a large part of the resource pie, and they're very closely tied to fitness. So we think about offspring size or number, growth, longevity, et cetera. Of course, these traits are composed, they stand on the shoulders of many other traits that sort of build up what we think of as growth or offspring number. And then there's a whole other large set of organismal traits that ecologists are often interested in because they are, we believe, that they mediate ecological interactions in some way and maybe adaptations in various ways. And in a way, this is going to be the heart of where I'm thinking and where this talk goes in terms of thinking about trade-offs. Nonetheless, since this is Stony Brook, I think it's important to give brief mention of life history evolution and in part how it's been so successful. Of course, Williams' theory of antagonistic pleiotropy in generating life history trade-offs was pretty successful. The idea, of course, is that mutations that are beneficial early in life but deleterious later in life do not get selected out of a population. This could lead to trade-offs between two life history components, such as fecundity and longevity. And Bobby and I were talking earlier about, I think, one of the examples that's quite controversial now in human evolution is this idea of estrogen being important for fertility early in life, but potentially having the downside of being associated with breast cancer later in life. And whether that actually will stand the test of time, it's unclear to me. But there are many of these sorts of life history trade-offs. Again, the kinds of traits we might think of in terms of life history, you can see right here. And what I want to do is transition your thinking to thinking about interactions that occur between plants and their animals. In particular, I work on herbivorous insects that eat these plants. And there's all kinds of justifications I could give you for why these are important species. Perhaps the two biggest ones is we're talking about the lion's share of macroscopic biodiversity. And in addition, essentially, all of biodiversity is getting its energy ultimately via the autotrophic action of plants. So in plant-animal interactions, the study of trade-offs has a long history in thinking about the evolution of defense. That is, if autotrophic plants are generating energy and herbivory is the conduit by which that energy is transmitted to the rest of the food chain, there's been tremendous selection over the eons for defense. And the hypothesis is simply that defense, as any other adaptation, must be costly and therefore exhibit some kind of trade-off. Those trade-offs aren't entirely obvious sometimes. In some cases, it may be due to a limiting resource that can only be allocated on the one hand to defense and to growth and reproduction on the other. On the other hand, other kinds of trade-offs that we tend to call ecological trade-offs may involve other kinds of negative associations. It may very well be that the defensive trait or compound, its energetic cost may be compensated for in a plant or in a particular genotype, but its expression reduces the efficacy or abundance of pollinators or other organisms that are interacting in a positive way with those plants. And in fact, there are now many research programs that are investigating those kinds of ecological trade-offs. There is both the energetic investment or trade-off and also the potential negative impact on other organisms. We heard some about multivariate strategies this morning, and I think there's long-standing hypotheses in plant-animal interactions about particular traits, when picked out of a hat, should negatively covary with other traits. But what in fact we all know, and this was discussed some this morning, is that traits make up complexes. And in fact, if we think about the alternative strategies that organisms have, they're composed of many, many traits. And those strategies themselves, or complexes of many traits, may trade off. And lastly, what I won't talk about, but what has a long history, and certainly one that Doug and Fred Gould have contributed substantially to, is the evolution of host range. That is, why are insect herbivores, so specialized in their diets? And the answer that we all want to believe is that jack of all trades is master of none. That is, that specialization occurs because of the increased ability to exploit a particular resource, but comes at the cost or detriment of being able to perform well on other resources. I'm not going to spend a lot of time on this, although I think it can be, it's certainly sort of part of the whole story. Measurement of trade-offs, of course, could occur on many forms, and one of the things I want to do in today's talk is span the micro to macro evolutionary perspective on trade-offs. As Resnick pointed out, trade-offs that were often measured as phenotypic correlations, that's fine. Those kinds of trade-offs may not necessarily be reflected in how microevolution proceeds. There are the middle category of genetic correlations or measurements of selection in alternative environments, again, that opposing selection. And then species correlations, which I think have enjoyed a dramatic resurgence in interest, in part because of comparative methods, comparative phylogenetic methods. We can now do more than compare two species and say that they have alternative traits, but we can, across many taxa, ask if a particular association has convergently evolved through Felsenstein's independent contrast or other methods. So what I want to do is just briefly go over with you some of the really nice examples from the literature on costs and trade-offs with relation to plant-animal interactions, and then I want to present some of my own data that I don't really understand yet, but I'm hoping that you can help me with. So I need to tell you a little bit of natural history about the way plants work in order for you to understand the rest of this talk. So this is the time to stare up from your computer screens and think about the incredible behavior in natural history that plants can exhibit. Most, if not all, plants produce a whole host of toxins, anti-nutritive compounds. Even lettuce is full of poisons. And remarkably, when plants are damaged by insect herbivores, a dramatic, highly conserved response is the upregulation of defensive traits. And those traits are hormonally orchestrated by a compound called jasmonic acid. That's not important. But the beautiful thing about it is we're talking about mosses and ferns and conifers and all the angiosperms. And we're talking about the same hormonal orchestration that will upregulate the production of nicotine, a toxic alkaloid in tobacco, that will regulate the increase in glucosinolates, the stuff that makes horseradish spicy. The same hormonal orchestration will increase the extrafloral nectar that exudes on acacia trees, or will increase trichome production as a physical barrier to feeding. We're talking about a very highly conserved immune-like response that plants have to defend themselves following attack. Now, It should be no surprise that organisms can't do everything, and there's a long-standing hypothesis that one of the reasons why defense is inducible or is upregulated following damage is it's a cost-saving strategy. And a classic example of working on the costs of production of those compounds comes from wild tobacco studied in the West, Nicotiana attenuata, and the herbivores. In this case, it's a hawk moth, which feeds on it. And the data I want to show you here comes from Ian Baldwin's lab, where he grew plants hydroponically with a limiting resource, nitrogen, which is limiting for their growth, labeled in the hydroponic solution, and simply phenotypically manipulated those plants to produce very little nicotine, higher amounts of nicotine. We're talking about a neurotoxin here, no known physiological function. We're talking really a defense or a high level of nicotine. And the amount of that labeled nitrogen that ends up in the nicotine pools in the plants increases substantially with that induction. Not surprisingly, most plants are limited in their lifetime seed production by nitrogen. And so if you look at the lifetime seed production in these same treatments, we see a decline. In other words, pretty strong evidence that we have a limiting resource, and there's a whole bunch of other data that I'm not showing you, that when it is phenotypically allocated to defense on the one hand, it is simply not available, cannot be recycled apparently, and not available for seed production. There are many examples of costs of defense that come from quantitative genetic studies and from selection studies. I'm going to just show you a cartoon here because the quantitative geneticists don't like graphs, and so I didn't want to show you a really boring table. But work from May Berenbaum, she's not here, so I can show some of her results, or Mark Rauscher's lab, has consistently shown, if we think about the furanocoumarins, which are toxic in the apiaceae that she studies, or the glucosinolates that Rodney Mauricio studied, what we see is that there is selection for defense in the presence of herbivores, but that these exhibit costs in the absence of benefits. In the absence of herbivores, we see selection against defense. And some of these have been phenotypic selection studies, although these two basically also showed the negative genetic correlations between investment in those compounds and fitness in the absence of being consumed. Now, the last sort of classic bit of data I want to tell you about here, to me, really is indicative of what we might call that adaptive negative correlation. And it comes from a paper published by Eddie van der Meijden from Holland in Oikos in 1988. It was an instant classic on defense and regrowth, alternative plant strategies in the struggle against herbivores. And the logic is basically that organisms in general, although they originally applied this to plants, may have two alternative strategies in the struggle against parasites. And the one is resist them. That is, evolve compounds or strategies that reduce the amount of parasitism. And the other is tolerance. That is, evolve strategies that minimize the fitness impact of those parasites. And certainly true for plants in addition to animals. Resistance to damage is predicted to negatively covary with tolerance in the case of plants or regrowth. And why? The idea is that very highly tolerant organisms, those for which the parasites do not reduce fitness, do not experience selection for resistance. On the other hand, those organisms which are highly resistant, which are not receiving damage or parasitism, do not experience selection to tolerate those parasites. And if these two traits are at all costly, then we expect that negative correlation to be expressed. And I say that this has been a huge success story in terms of predictive adaptive negative correlations in plant ecology. It's been shown intraspecifically across taxa, and the animal literature has picked up on this. There was a paper in Science last year, I believe, on mice or other rodents that showed this same negative correlation between tolerance of parasites versus resistance. Okay. What I want to do is transition to a little bit of natural history about a group of plants that I study, in which I'm trying to apply some of these ideas and disentangle whether we're seeing trade-offs versus adaptive negative correlations in the strategies that these plants have. So I'm going to tell you about a group of North American plants in the genus Asclepias. They take their name from the Greek god of medicine, Asclepios, in part because they've been used for thousands of years in medicine. They mess with the sodium-potassium pumps of animal cells, and in particular invertebrates, on cardiac function. It's a beautiful, diverse group of plants, 140 species in North America. There's something very Darwinian about this image to me because the floral form is quite distinctive for the genus, but of course what you can see in the close relatives is that the coloration and form has been stretched in various ways over the course of the radiation. This work we do intraspecifically on one common species, the common milkweed, which you have around Stony Brook here, and in addition in a comparative framework, working on about a third of the extant species, about 50 Asclepias species with my collaborator, Mark Fishbein. And the two traits I want to focus on are pretty interesting. One is latex. All of you have broken a dandelion or a milkweed plant or a fig leaf and experienced that milky substance. This is a highly convergent trait in plants. It's about 10% of plant species. It's about 10% of plant families. And you can just take that to indicate that it's likely to have evolved many times. It's a physical barrier to feeding. Latex is contained under pressure when the caterpillar or other herbivore feeds on the leaf. It's basically like getting a gallon of paint, toxic paint, thrown in your face. On the other hand, the other trait I want to focus on is the cardenolides, or cardiac glycosides, for which the genus of plants take its name. These are toxic steroids of terpene origin. The cardenolides occur in less than 0.1% of plant species and in about 3.5% of angiosperm families. The lion's share of species that have cardenolides occur in one family, the apocynaceae. We see a strong pattern of phylogenetic conservatism for that trait. The plants are really diverse. They grow in various wet and dry habitats, needle-like leaves, broad leaves. It's really a beautiful system to look at the diversity of form and function. These traits really matter for the herbivores. I just want to show you very quickly. If we look at a genetic correlation where each dot here represents a genetic family of plants, there's a high level of variation among genetic families, and that negatively co-varies with damage by this weevil. So these traits are important in terms of mediating ecological interactions. If we look at cardenolides, this is 18 species of Asclepias negatively co-varying with, in their cardenolide content with the Rmax of an aphid. The aphids in their feeding, of course, feed intercellularly and therefore don't puncture the leuticifers. They don't experience the latex, but they certainly experience the cardenolides and the phloem and are negatively impacted. I should say both of these herbivores, as all of the herbivores of milkweeds, are specialists, and yet they still are, they're dealing with the problem because presumably they're engaged in some coevolutionary interaction. Although they're specialized, they have their mechanisms to deal with these toxins, they're still negatively impacted by them. Okay, so a long-standing hypothesis, which in hindsight to me seems trivial, is do defensive traits trade off? That is, do latex and cardenolides, two potent defenses of these plants, show a negative association? And I say that it seems trivial now to me in retrospect because there's no a priori reason that they are ecologically redundant traits, although I should say that there are reviews and meta-analyses in evolutionary ecology asking whether adaptive traits like defenses negatively co-vary in the absence of that a priori prediction. Typically, they find in the case of defenses that there is no trade-off if you arbitrarily pick two traits out of a hat and ask do they negatively co-vary. If you look at latex and cardenolides across 20 genetic families of milkweed, there is no trade-off or no negative genetic correlation. These are data from 3 different environments. What the environments aren't important, but you see that they basically are flat. If you look across 35 Asclepias species in a comparative phylogenetic analysis, there is no relationship between latex exudation and cardenolides. And so I guess what I would say is that if this is the wedge of resources, limiting resources for defenses alone, it's relatively small, remember, in the large scheme, and plants have many, many forms of defense. There's no reason to expect that 2 of these trade off. In fact, we find no pattern of a trade-off. And it very well may be that there are negative associations if we think more in terms of the functional aspects of redundancy between the kinds of traits that these organisms have. What I want to turn to in the last 5 minutes, and this is really the heart of the talk. I'm going to talk fast now. is the deployment strategies. I'm going to focus on cardenolides. There's a long-standing prediction in plant-insect interactions that the inducibility, remember, these plants upregulate defense in response to herbivory, the inducibility or amount of increase after damage should negatively covary with the constitutive resistance, with that is basally or allocated to without any damage. And one question we can ask, is this an allocation trade-off or an adaptive negative correlation? Now, the work I'm going to present was collected by these various folks. And to cut to some of the data right here, we're looking at 20 genetic families, constitutive allocation to cardenolides, and the amount of induction or increase. There's a very strong, remarkably strong negative correlation. Those genotypes which are very high basally, in fact, decrease in their cardenolides after damage. Those that start out low increase a lot. We can look at that same exact data as a reaction norm plot. These are the same data. Those that start low increase. Those that start very high decrease. Now, one interpretation of this is that this species has a maximum amount of cardenolides that it allocates to, that it can acquire and allocate. And it's simply deployed in two different ways, as a constitutive strategy or as an induced strategy. What we don't know the answer to is this sort of hypothetical genotype possible? That is there truly an upper limit, in which case there's a physiological trade-off or constraint in terms of allocating resources, or is it the case that simply this phenotype is never favored? That is, it may be that genotypes that allocate a lot constitutively, if they're receiving damage, more cardenolides is not going to help. And so this negative correlation between constitutive and induced allocation could be an adaptive negative correlation. As a way to try to understand how plants acquire and allocate these defenses, one of the things I did was try to ask, what is the pattern across taxa? What is the macroevolutionary pattern across taxa? I'm going to show you two data sets here. And the first is basically looking at 12 species of Asclepias. The cardenolides that they produce are on the y-axis. They are arrayed in terms of their constitutive or basal undamaged amount of toxin produced. When you damage them, if there was a trade-off, you'd expect the greatest induction here and the least induction there. What we find is exactly the opposite across taxa. The 4 species which show significant induction and which increase the most basically occur on this end of the constitutive spectrum. No trade-off or positive correlation. We just have analyzed the data across 52 Asclepias species. This is an incredible experiment. Monarchs naturally feed on all of them. There is a positive correlation in a phylogenetically independent contrast across the initial level of investment and the amount that they induce. So what we have here is a countervailing trend of within species trade-off, across species positive correlation. What I'd like you to think about is what is going on here. And I just have a hypothesis to propose, is that we have that same relationship, constitutive defense, induction here on the y-axis. Asclepias syriaca, where we saw that trade-off can be represented by one of these colors. Let's call it these black lines. Within a species, we see that this relationship is negative. I would hypothesize that is likely due to the resource limitation in terms of acquisition and allocating to cardenolides. And the reason I make that sort of speculation is that as taxa evolve and speciate, what we find is that they vary tremendously in the amount of cardenolides that they make and allocate, but that is proportional to what they can allocate to the induced defense. So it appears that basically there is this upper limit to what species can make. I'll simply add, I don't have time to really discuss this, but during the adaptive radiation of Asclepias, what we find is a directional trend decreasing in the amount of cardenolides that these plants make. And we see that an increase or an escalation in the tolerance during the diversification process. And so I have 0 time now. I'll leave you with this and these conclusions. Thank you very much.